Hello, my name is Megan Callahan Gillespie, and I'm here today on behalf of Dr. Mark Manary and the Manary Research Lab at Washington University in St. Louis. Today, we'll be talking about a quick start project on a cowpea biomarker study that was conducted in Ghana. First, I want to start with a little bit of background information for the rationale for this project, which is to move nutrition, specifically cowpea nutrition, into the 21st century. You'll see I have a quote on the slide that says, "No science achieves maturity without precision data." Nutrition is the science of food, but we can't reliably measure food intake. In order to interpret the effect of diet or food on health, it is critical to accurately measure an individual or a population's dietary intake. Today, we often use self-reported dietary assessment methods, which include food frequency questionnaires, dietary recalls, weighted food records. These methods have so many limitations. They're high in cost, they're time-consuming, they involve participant recall bias, there's measurement errors, there's often misreporting or underreporting of dietary intake, and difficulty determining accurate portion sizes. Just imagine trying to understand diabetes without being able to accurately measure blood sugar. This is what we're talking about when we say we need precision data. We need a reliable measurement for dietary intake. The discovery of dietary biomarkers is an area of increasing interest. Today, there are only a few biomarkers for dietary assessment that are well established. This study has the potential to benefit future nutrition and education-related programs, policies, and research across multiple disciplines by offering a quantitative measure of intake with the identification of a cowpea biomarker. This precision data will reduce the time, effort, and inconsistencies related to subjective dietary intake methods, and most importantly, advance our understanding of the relationship between diet, or in this case, cowpeas, and health. So you might be wondering, what's the deal with cowpeas? Well, in 2016 and 17, after we conducted a study in rural Malawi among infants who received one of 3 daily supplements, a common bean flour, a cowpea flour, and a corn soy blend control, we found that the cowpea supplement resulted in less linear growth faltering. This was encouraging because up until this point, there had not been interventions to show a reduction in stunting in the context of rural Africa. And because cowpeas grow well in Africa and they're culturally accepted, plus they're a hardy, drought-tolerant crop, we had even more hope that our findings may provide an affordable and accessible solution to vulnerable children in Africa. But since cowpeas are grown mostly in the African continent, we know that they have not been studied as extensively as legumes in North America, such as soy. So this is novel work that will be vital in linking cowpea consumption to human nutrition. So our quick start proposal was titled Measuring Cowpea Consumption in Infants and Pregnant Women in Ghana. And as I said before, the study aim was a reliable and convenient way to measure the quantity of cowpea that was being consumed by an individual. And our objective was to identify a novel set of dietary biomarkers that will measure cowpea consumption and be free from participant recall bias and serve as a way for us to quantify this legume intake. In other words, our objective was precision data. So now let's move into the methods. First, I want to talk a little bit about our study timeline. All of the study data that was collected in the field was collected between August and September of 2019, so pre-COVID. All pictures are from pre-COVID times. We had 40 study participants enrolled into the study, 20 children between the ages of 9 and 21 months, and 20 pregnant women that were older than 18 years. Participants consumed measured amounts of cowpeas in increasing quantities, starting with a period of no cowpeas. I'll talk about this in a little bit more detail on the next slide. We collected urine samples and blood spots, and urine samples underwent metabolite detection via ultra-performance liquid chromatography tandem mass spectrometry. Here you will see an outline of the 4 5-day phases of the study. As I said before, we started with a washout period where no cowpea was consumed. This was followed by gradual increases in the amount of cowpea consumed throughout the remainder of the study. Both pregnant women and infants came to a common location every day during the study period to either answer questions or receive their pre-portioned amount of cowpeas. This way we could observe that the cowpeas were being consumed and record it appropriately. So now we're going to start looking at the data. So we'll start with the PCA plots of the foods that were consumed. We have 4 cowpea varieties that we use for our biomarker study, and then we also have navy bean on this plot for comparison purposes. And this is the metabolite profiling done on the foods before they're consumed by a human. So these are the molecules that you would find in the food ingredient themselves. And you can see the purple up there is the navy bean by the legend. And then there's a cluster of 3 of the cowpea varieties down at the bottom. And then on the left, you have the orange, which is another cowpea variety. And this is revealing that there's molecular differences between legume types, obviously between the cowpea and the navy bean, but there's also differences even within the same legume and the different varieties. So with the 3 cluster cowpea varieties at the bottom, we see that we would likely have some overlap there, and there would be something to identify cowpea in general. So here we are looking at the differences between children and women, and we have PCA plots of all diets and all dying points. And what we want to show here is that when we look at all of the small molecules found both in pregnant women and children, we have 2 distinct sets that are found specific for the child or the pregnant women. So you can see the red is for the children and the green is for the pregnant women. So this is not so much unexpected, as we know that the metabolic response to any dietary intervention among these populations is going to be a little bit different, given that they're going through different things and are in a different physiological state. But we were hoping that we would have one universal magic biomarker. But since we don't see that the colored circles are overlapping, it's likely that the biomarker won't be there for both pregnant women and children together. So now we're going to look at the urine metabolomes in 2 different time points, the washout and the high consumption, and we're going to look at women and children that are analyzed separately. So here we're looking at the PCA plot of the washout period and the high consumption period for 2 of the cowpea varieties, the Dagbantua and the Sangui. And because I don't want to continue to mispronounce those, we will call those the DS variety. In red, we have the high consumption, and in green, we have the washout. And what we're looking for here is to see if the small molecules found in the urine during the high consumption period are different than in the washout period. And we are looking at that in both children and in pregnant women. And this next slide is very similar to the previous slide, except that we're looking at two other varieties of cowpea, the Tucara and the Adua, or the TA varieties. But again, we're looking at women, pregnant women and children separately, and we have the high consumption as the red and the green as the washout period. And we want to see if there are small molecules found in the high consumption period that are not found in the washout period. So we're looking for the lack or the absence of overlap. Here we have another visual looking at shared metabolites when comparing the urine molecules found in those that consumed the different varieties of cowpeas. We see that we're comparing and seeing if there's overlap between the pregnant women and children. And we see with those that consume the DS varieties, there are 2 common elements. And among those that receive the TA varieties, there is 1 common element. So now we have candidate cowpea biomarkers. First, we have our cowpea general consumption biomarkers. And this category includes phytochemicals unique to plants, such as quinic acid, other general biomarkers that tell us what the effect of cowpea is in human metabolism, such as carnites, which are antioxidants and necessary for energy transfer. And this gives us a little insight into how cowpeas strengthen human nutrition. The variety-specific biomarkers are useful if we wanted to test a certain variety that was being consumed, or perhaps there was a new variety, or we had a variety that we knew had a specific advantage or benefit. Now, you might be thinking, aren't there thousands of different small molecules in cowpeas? Keep in mind that PCA plots, which is what we've been looking at, just tell us the difference between people consuming cowpea and people not consuming cowpea. And while thousands of different molecules are put into the gut by eating cowpea, there are not thousands of molecules in urine to detect. This is because resident in our gut are a multitude of microorganisms that eat up any organic matter that's not absorbed by the human as nutrients. They transform this organic matter to functional biological molecules. This is a very efficient way to fully utilize the food that's being consumed. It also makes looking for a biomarker kind of difficult, as it cleans up all the traces, or what we thought would be the traces, of cowpea and what we hope to detect. So while our first idea was that we would find something unique, like a molecule in a cowpea that wasn't in any other plant and was stable and appearing in the urine, no such molecule seemed to exist. But that doesn't mean we weren't successful in identifying a biomarker. Here you'll see a figure with orange and blue bars, one for pregnant women and one for children. The orange represents the high consumption diet, and the blue represents the washout. The biomarker that we discovered is a set of small molecules that change in a certain coordinated pattern with respect to each other. This is a suite of molecules, and it replaces our original desire for a unique singular molecule. You'll see it here on this slide for the TA varieties, but we also found it in the DS varieties. So we have both biomarkers for both pregnant women and children, and biomarkers based on the different cowpea varieties. As I'm just about to hit my time requirement, I did want to make sure I had a few closing remarks. First, the pairwise comparisons for both pregnant women and children within each diet did demonstrate a suite of metabolite biomarkers for cowpeas. This is a huge success. Our next step will be to quantify these biomarkers in a larger sample set with commercial standards. Unfortunately, currently, with the situation outside of our control due to COVID, commercial standards have been hard to come by. But I'm not worried. I know that as soon as we can get our hands on those commercial standards, we'll move forward with our next steps. And finally, I wanted to say we will achieve the goal of precision nutrition. But it's going to require continued efforts to develop, validate, and refine assessment methods that can capture the multidimensional nature of the human diet. And finally, I wanted to make sure to acknowledge all of those who this project would not have happened without, especially those at the University of Ghana, Professor Steiner Asidu and Professor Salia, as well as our collaborators at Colorado State University, Elizabeth Ryan, of course my colleagues in the Manary lab at Washington University, and without the generous funding from USAID, Global Food Security, and the ongoing support from the Feed the Future Innovation Lab for Legume Systems Research. We appreciate all of our collaborators and collaborations. Thank you.